Hello everyone. Let us look at gluconeogenesis. Gluconeogenesis. From the name, gluconeogenesis, synthesis of glucose from the non-carbohydrate precursors. It takes place when the level of glucose is low. We are talking about at the point of starvation. So we have to agree that when the glucose level is high, the hormone insulin will regulate the glycolytic pathway. When the glucose level is low, glucagon regulates gluconeogenesis. It means these two hormones work antagonistically. So when the glucose level is high, insulin as a hormone will try to bring it to the normal level. But when it's low, the glucagon has to fight to look of ways to bring it to the normal level. So those two hormones are very important in blood sugar regulation. So by definition of gluconeogenesis, we are synthesizing glucose from non-carbohydrate precursors. And the most important thing here is all cells depend on glucose, especially the brain. So glucose level in blood plasma must be stable because all cells depend on glucose. So the brain, for example, it is very sensitive when the glucose levels decrease because it can lead to impairment of the brain function. The red blood cells also use glucose as a fuel. So during starvation or during fasting, glycogen is always used as a source of glucose, but it's not enough. Glycogen stores glucose. Storage for glucose is always not enough. So the gluconeogenesis will come handy because it will be able to synthesize glucose from the non-carbohydrate precursors. So that is the most important thing, because the glycogen stores, when they are not enough, they just provide glucose which lasts for 12 to 24 hours. Then the body will look for other ways of synthesizing glucose from the non-carbohydrate precursors, and that is how definition of gluconeogenesis is. So these non-carbohydrate precursors are known as the lactate, pyruvate, glycerol, and some of those amino acids. We saw one when we are looking at the amphibolic role of glycolysis. We talked about amino acid like alanine can be synthesized from pyruvate. So in that case, alanine can also be one of the amino acids which also used much in the synthesis of glucose when you talk about the non-carbohydrate precursors. So liver and kidney are the major sites for glucose synthesis, and under fasting conditions, then gluconeogenesis as a pathway has to supply the body with glucose. Most people think gluconeogenesis is a reversal of glycolysis. No, it is not a reversal of glycolysis. What we are doing is, if the arrow is going towards pyruvate in glycolysis, because by definition you are defining glycolysis as breakdown of glucose to pyruvate with the main objective of generating energy, so the arrow goes towards pyruvate. But in gluconeogenesis, you are synthesizing glucose from the non-carbohydrate precursors. So the arrow should go towards glucose formation. So necessarily is not reversal of glycolysis, but we had 3 irreversible steps in glycolysis. Step 1, which was catalyzed by the hexokinase. Step 3, where the phosphofructokinase was, and step 10, where the pyruvate kinase was. So those 3 steps are bypassed, but all the reversible steps remain the same, as long as the arrow goes towards the synthesis of glucose. So in this case, we are talking about the bypassed steps. The bypassed steps are the 3 irreversible steps in glycolysis. We are going to bypass them. And these bypass steps, you can see the step 1, 3, and 10. They are the bypassed steps in gluconeogenesis. So if a step in glycolysis was a step number 1, it is going to be the last step, or step number 10 in gluconeogenesis, because now the arrow goes towards synthesis of glucose. So the first bypass involves 2 enzymes, because there is an intermediate which come in there. So we are converting first pyruvate to oxaloacetate, then from oxaloacetate to PP. You know, in glycolysis, it was 1 step from PP to pyruvate. But here, there is an oxaloacetate in between. And oxaloacetate is 4 carbon, pyruvate is 3 carbon. So that is carboxylation. So in this case, we talk about pyruvate carboxylase as the enzyme which converts pyruvate to oxaloacetate. Then we have phosphoenol pyruvate carboxykinase, which converts the oxaloacetate to phosphoenol pyruvate. So that is bypass 1. Then we have bypass 2, where the PFK was. It was forming fructose 1,6-bisphosphate from fructose 6. In this case, the fructose 1,6-bisphosphate is converted to fructose 6-phosphate. It means in glycolysis it was a kinase. Now it is a phosphatase. Phosphatase removes a phosphate group. Kinase adds a phosphate group. So the enzyme, being the substrate is fructose 1,6-bisphosphate, the enzyme is fructose 1,6-bisphosphatase. The last bypass, or bypass 3, is the formation of glucose from glucose 6-phosphate. So the enzyme is glucose 6-phosphatase. It involves formation of glucose from glucose 6-phosphate. If you look at this diagram, it shows gluconeogenesis. So the red colors, is it red or pink? Pink colors shows the bypassed steps. But the blue ones were the reversible steps in glycolysis, which remains the same in gluconeogenesis. So what it shows there is we are defining synthesis of glucose from the non-carbohydrate precursors. We are synthesizing glucose, so the arrow goes towards glucose. So the non-carbohydrate precursors, we talked about lactate. Lactate here, where the lactate dehydrogenase is going to form pyruvate, then it goes to synthesis of glucose. Some amino acids like alanine, which we defined in the previous slide in glycolysis, are going to be converted to pyruvate. Then they can go towards formation of glucose. Some amino acids can connect at oxaloacetate. These amino acids, example, are the aspartate, which is going to connect to oxaloacetate, then be converted to glucose or be used in the synthesis of glucose. The other one is glycerol through the dihydroxyacetone, which was one of the trioses generated by aldol cleavage. So the glycerol will be converted to dihydroxyacetone phosphate, which will further be interconverted to glyceraldehyde 3-phosphate. The 2 will form fructose 1,6-bisphosphate and further will form glucose. So the pink ones show the bypass step, but the blue ones remain the same because they are the same enzymes which were seen in glycolysis. This is the first bypass. We have pyruvate, which is 3 carbon atom, carbonic acid, which we have the bicarbonate present there. It's going to form oxaloacetate. The enzyme is pyruvate carboxylase. The enzyme is ATP dependent, or it used there ATP as a source of energy. So in this case, this reaction takes place in the mitochondrial matrix. I said, when I was doing the introduction to Biochemistry Simplified, I said a cell is the basic unit of life. And if you talk about the basic unit of life, you have to define biochemistry, the chemistry of life. And what is life? If something, reactions are not taking place, reactions have to take place. But they have to take place where? In the organelles, the subcellular components. So already there is an organelle, mitochondria, the reactions which take place there. The organelle, the reaction which is active. When we talk about the organelle, we have to relate with the cell, the functions. So it's very important to appreciate biochemistry. It is not as difficult as people think, but we have to make sense out of it. And that is why we are trying to simplify the biochemistry, so that the students who already thought it is hard, they can see a way which gives them hope, so that they can try to read and understand and pass. So this takes place in the mitochondrial matrix. Still on bypass 1, but now oxaloacetate is converted to phosphoenol pyruvate, the PEP, phosphoenol pyruvate carboxykinase. This takes place in the cytosol, just the same way as glycolysis was taking place in the cytosol. This reaction takes place in the cytosol. There is decarboxylation of a 4-carbonoxaloacetate to phosphoenol pyruvate, and the enzyme is involving phosphoenol pyruvate carboxykinase, removal of one molecule of carbon dioxide. The enzyme is GTP dependent. Second bypass, fructose 1,6-bisphosphatase. This is an a hydrolase, sorry. So water is involved, a hydrolase converting fructose 1,6-bisphosphate to fructose 6-phosphate. So the enzyme, with addition of water, removes one of the phosphate from the first carbon atom to form fructose 6-phosphate. Third bypass, formation of glucose, and this takes place in the ER. So in this case, we talk much about different organelles or the subcellular components where the steps of the reactions take place. So the formation of glucose from glucose 6-phosphate, this step does not take place in the cytosol, it takes place in the endoplasmic reticulum, and the enzyme is glucose 6-phosphatase. We are removing a phosphate group from glucose 6-phosphate to form glucose. Regulation of these enzymes, very important. Insulin as a hormone will regulate glycolytic pathway and will also regulate glycogen synthesis. Glucagon, for example, will regulate gluconeogenesis and also will be regulating glycogen degradation. So at that point, insulin is regulating glycolysis. It is also stimulating the expression of the enzymes which catalyze the irreversible steps in glycolysis. So it stimulates the expression of hexokinase, phosphofructokinase together with pyruvate kinase. Glucagon, on the other hand, when insulin stimulates this enzyme, glucagon will suppress its enzyme because I said they work antagonistically. So glucagon rises during starvation and it will inhibit the expression of enzymes which are involved in glycolytic pathway. But it is going to stimulate the production of the enzymes involved in the bypassed steps.